Have you ever wondered what makes Michael Phelps the best swimmer, or Usain Bolt the fastest sprinter? Why some people just seem like they were born to be an athlete? What if I told you they were born like that? That it's literally in their genes? Discoveries of common genes that can predict success in sport is nothing new, but 6 genes were found to potentially predict the traits of an Aussie Rules football player. Do you have them? Welcome to the genetic basis of stuff and things.
A 2012 study claimed that 66% of variance in athletic status could be explained by genetic factors.
Your genes determine, to an extent, what your physical characteristics will be, your phenotype. This can be altered by epigenetic contributions or environmental factors. Success in sports has multiple contributing factors, but a person with a favorable genetic profile and in an optimal training environment is seemingly bound to be an elite athlete.
There are certain traits that are favorable in almost any sport, such as
Endurance, muscular strength, and trainability. All of these can be influenced by genes, potentially these genes, but we'll come back to these later. There are over 200 polymorphisms that have been associated with physical performance, and at least 20 of these have been scientifically associated with elite athleticism. So, which of these are of interest for Aussie Rules? A 2019 study conducted by Chivers and Anderton found that there were 6 genes of interest when it came to sub-elite Australian football. 30 players were genotyped and tested in abilities on a 3x1 kilometre time trial and direct game involvements per minute. This includes handballing, kicking, and marking. The following genes were found to determine success in direct game involvements, and these for the running time trial. It was evaluated whether there were significant associations between specific genes and performance in these tests. This can be seen in p-values, identifying these polymorphisms to be significant predictors of the 3x1 kilometre time trial, these for handballs, and this for kicking assessments. But why? All 6 genes can be mapped out in their relation to the activation of CREB, a regulatory protein in charge of cell differentiation, survival, and proliferation. PPRGC1A is involved in endurance. This is because this gene plays a large role in glucose and lipid metabolism, as well as muscle fiber formation. ACE is associated with increased endurance, strength, and power, and is the most widely studied gene for human physical performance. It is responsible for controlling blood pressure through regulation of blood fluid, sodium and water levels, as well as circulation. This ensures oxygen flow to muscles, as well as the regulation of heart function. COMT encodes for an enzyme which promotes the breakdown of dopamine. A study involving rugby players found that elite players were associated with lowered levels of this allele and could improve resilience to stress, better performance in competition, lowered concussion risk, and faster recovery time. The ADRB gene increases maximal oxygen consumption and endurance. This is because these receptors are involved in the regulation of cardiac function and muscle relaxation, as well as the mediation of adrenaline responses. A Korean study involving college athletes found a positive relation to those with ADRB polymorphisms and athletic success. DRD2 is responsible for attention, memory, sleep, and learning through the regulation of dopamine synthesis, storage, and release. A 2021 Turkish study suggested that the DRD2 gene can be associated with addiction and thus competitive behaviors. This is likely due to reward systems in the brain. The gene was positively associated with athletic performance of football players. Lastly, BDNF is a gene that is commonly associated with short-term motor learning due to motor cortex plasticity. This is the ability for neurons to learn from previous activities and modify their response. This has a key link to the trainability of an individual. So what? What is the benefit of identifying these genes? This study found that genetic studies in sports can have 2 key benefits: athlete selection and athlete training. The current screening methods for AFL is a standing vertical jump, running vertical jump, 20-meter sprint, agility test, and a 2-kilometer time trial. Genetics pose the potential for a novel method for seeking player ability and a way to diversify selection processes. However, selection solely based on genetics is not only naive, but maybe even unethical.
In 2019, an Australian study suggested a 5-level performance assessment model to be utilised in Aussie Rules Football.
First, a laboratory test, then a field-based static test, field-based dynamic test, small-sided game test, and finally match play. This offers a more well-rounded selection process, acknowledging the genetics and training of the athlete.
Genetics can also be used to improve training by tailoring it to the genetically predisposed talents of the player.
For example, in AFL, the midfielders run the furthest and do the most handballs, whereas forwards do the most kicking and most sprinting. Thus, by assessing the genetics of the player, they can not only be placed in the optimal position in the team, but can train to improve their predisposed talents.
Limited studies have shown the same results in what genes make an athlete,
likely due to different traits required for different sports. So far, only Jacob has done genetic research in Aussie Rules Football, publishing another article in 2023 on genes predicting injury susceptibility. She is a pioneer in Australian genetics, advancing our knowledge on our beloved sport. However, it is clear that these 6 genes play huge roles in Aussie Rules Football and should be further researched. So, were you born to play Aussie Rules?